Good evening, everybody. Hope all participant in. So we should, because it's already 4, so we should start. So first, welcome to all of you at One Cell seminar or webinar series. Today we have invited Dr. Shalini Bhattacharya for she is more on the cancer stem cell, and she will highlight the cancer stem cells and importance and new advancement, and also their role in how to design or how to go for the therapy path. So, so Dr. Bhattacharya is heading of the Department of Biophysics in PGI-MER. It's a well-renowned medical and research organization in India since 2016. So Dr. Bhattacharya and her team working on the cancer stem cell, and especially on the EMT, that is the epithelial-mesenchymal transition, which is during the cancer, especially in the renal cancer carcinoma or urinary bladder carcinoma. She is a recipient, or she received many national, international awards, including the Young Scientist Award by the Indian Science Congress. So not taking much time, I would say the primary objective of this webinar is to primarily focus on the latest development on the cancer stem cells, which is the keystone in the development of cancer therapy and treatment option in future. So taking not much time, I would request Dr. Bhattacharya to start her valuable talk, and we'll take the questions at the end of the talk. So please, can you start? I will switch off the camera at this moment, so we'll be easily audible. Thank you.
Thank you, Dr. Lippi. Thank you, Dr. Lippi, for your introduction. I'll now share my screen. So I think okay. Is this visible? Is the slide visible?
Is it visible, ma'am?
It is visible. So
yes,
as Dr. Lippi said, so I'll be speaking on stem cells to cancer stem cells and their role in cancer therapy. So cancer is a disease which has been there with mankind since time immemorial, because even in the Egyptian mummies, it has been found that they had cancers in their bodies. So probably mankind has been trying to find a cure for cancer since that era. Over the last few decades, we can say that we have improved the cancer therapy with new technologies, advancements in science and technology. We have made a more personalized series of drugs or personalized therapies, which can also take into account the genetic profile of the individual, or as well as the cancer profile of the individual. So in this context, I will be talking about how the identification of a subpopulation of cancer stem cells has led to a paradigm shift in the cancer therapy. Though we've not been able to eradicate cancer completely, but still the world statistics show that the mortality has decreased. We have many cancers which can be controlled, and survival period have increased, and quality of life of cancer patients have improved over the past decade. So in this webinar, I'll be talking about how the cancer stem cells give rise to, how the stem cells give rise to cancer stem cells. And after that, I'll be talking about some of the in vitro studies we are carrying out in our lab. I'll share some of the data from my lab with you regarding how an in vitro model of cancer stem cells can be generated to try out different drugs, to look at the pathways to improve the existing therapy. As I said, cancer is accumulation, is a disease that is caused by accumulation of genetic abnormalities, resulting in production of a cell which has unlimited proliferating capacity, leading to invasion to the other organs, as well as to the other parts of the body, leading to a metastatic disease. So the whole process of carcinogenesis somehow resembles the renewal and differentiation of the normal cancers or of the normal stem cells. Just as differentiation and proliferation are a part of the normal stem cell physiology, in tumors also, what happens is this differentiation, this control over the self-renewal and differentiation goes haywire. As a result, the cells, they become either de-differentiated or they undergo a maturation arrest. And that can occur at any stage of the stem cell proliferation or differentiation, including in the adult progenitor cells, in the differentiated cells, or even in the de-differentiated cells. So accumulation of all these mutational events can finally cause the cancer or the tumorous cells to divide and without any control. The concept cancer arises from the stem cell is not new. Way back in 1855. Pardon? Somebody please switch off your audio.
Yeah, I'm requesting all the audience can mute.
Yeah, thank you. So it was way back in 1855 that the embryonal rest hypothesis was given, according to which the cancers arise from tissue stem cells in adults. So even microscopic examinations of the tumor samples revealed presence of embryonic stem cells, which are akin to the developing fetus. Another example is the teratomas. So teratomas are non-malignant tumors comprising of a heterogeneous population of cells. So as I have shown in this image, this is a teratoma which comprises of a ball of cells. So in these cells, there is a teeth. So basically, it's a combination of different embryonic stem cells which have gone haywire. So they are stopped at different stages of differentiation. And as a result, at that point, they start dividing. So they differentiate and divide and give rise to this mass. So they can divide into any of the cell types. And combining all this, the key cellular event in the progression of cancer is first you have a series of mutations that eliminate the restraints on the cell division. Otherwise, a cell is expected to undergo a very disciplined division. You have read about cell mitosis, cell meiosis, so it's very well regulated and well documented. But when mutations occur, combination, it's not a result of a single mutation, there has to be a number of mutations that occur together in the cell. As a result, these restraints in the cell division is lost. And the cells start proliferating or dividing indiscriminately. And the cells lose their organization, and as a result, they enter into the angiogenic systemic circulation. So I'm talking here mainly about the epithelial tumors. So once they enter into the systemic circulation through the angiogen, they travel to the other organs and they spread through the blood vessels and the lymphatic systems. And this is the stage what is called the metastasis. So the primary tumors from the primary tumor site, they travel to another site, and then that is called the metastasis. And wherever they find a conducive environment, they get established. So they form the secondary tumors. Then you have the secondary tumors and the tertiary tumors, and as a result, these tumor cells spread throughout the body, leading to indiscriminate cell division. And as a result, they take over the cell division machinery, and the normal cells are overcome by the cancerous cells and leading to a multi-organ failure. So the tumor population doesn't comprise of a single type of cell. It's a heterogeneous population. So the difficulty in treatment of tumor is because of the intra and inter-tumor heterogeneity. So inter-tumor heterogeneity means the same within the tumor cells, there are subpopulations of different types of cells which need to be targeted to completely eradicate the tumor. And inter-tumor heterogeneity means that, say you have a breast tumor. So there are so many forms of breast tumors. Breast tumors in all forms are not same. So they cannot be treated by the same type of drugs, or they do not have the same targets. So again, they have to be profiled. So there comes the role of the personalized therapies. So you have to profile that which kind of receptors are upregulated. So according to that, you can design your drug. Over the period, different models of tumorigenesis has been proposed. The earliest model is the stochastic model. According to this, when a mutation occurs in a cell, it loses its proliferating capacity, and as a result, it continues indiscriminate divisions. So this leads to the formation of a tumor. Then came the cancer stem cell model. According to the cancer stem cell model, the stem cell or the progenitor cell can undergo a mutation, leading to, again, indiscriminate division of the stem cell or the progenitor cell. But both of these theories are good by themselves, but they not express the whole scenario or the whole profile of the cancer. So for this, both the theories have been collated to form the CSC plasticity model. So according to the CSC plasticity model, the mutation can occur at any stage because of the genetic or epigenetic factors. So they can be at the level of stem cells or they can be at the level of the differentiated cells. And there is also a possibility that a differentiated cell gets de-differentiated. It gets de-differentiated and then forms a stem cell-like phenotype. So therapy model for each type will need to be recalibrated and designed accordingly. This cancer stem cell theory over the period has emerged as an attractive theory because it can explain many of the unsolved queries regarding the cancer proliferation and treatment. So by definition, cancer stem cells is a small subpopulation of neoplastic cells which grow in an anchorage-independent manner. They show pluripotency and they are more resistant to the drugs. They can explain the whole idea of tumor heterogeneity because if you have a stem cell-like cell within the tumor population, it can give rise to any type of cells. It can form a mesoderm, it can form an ectoderm, it can form a endoderm. So that is what the tumor heterogeneity is all about. It does not comprise of a single type of cell. Again, the inefficiency of metastatic formation. Now, all the tumors are not actually metastatic. So the tumor also need to work hard to move from its initial site to the secondary site to metastasize. So only 1 to 2% of tumors actually metastasize. So one of the reasons can be because the cancer stem cell population within a tumor is very low. It's 0.1 to 0.5%, which is a very small population. Again, it can explain the tumor regrowth after an apparently complete remission because when these drugs are given to ablate the tumor, it's mainly targeting the proliferation mechanism. So it is targeting the DNA where the divisions is taking place. So only the fast growing cells are targeted. However, the cancer stem cells are more quiescent. They don't undergo proliferative divisions very fast. They only divide when they require, when they get sufficient proper microenvironmental signals. So that is why they remain quiescent for a long time. And once the main tumor bulk has been removed by the chemoradiation or chemo or radiation methods, the tumor remains in a dormant state for some time, and the cancer stem cells, they survive. So after a certain period of time, whenever they get a conducible environment, they can be triggered. So you can see that after 4 or 5 years of remission, the patient can again develop a very aggressive form of cancer. And the cancer stem cells can also explain the concept of drug resistance because these are the quiescent cells. They have a better drug efflux mechanism. So that is why the drugs don't act on them. So we'll be coming to that later. What leads to acquisition of the cancer stem cell phenotype? It has been observed that the cancer CSC phenotype can be achieved when there is a reactivation of the developmental pathway. When a cell differentiates, in a stem cell, the developmental pathways like Wnt, Notch, Hedgehog are activated. But as the cell differentiates to a particular lineage, these signals slowly mute down and are almost lost in an adult cell. However, in case of cancer stem cells, these signaling pathways are reactivated. So the differentiated cell is transported back to a de-differentiated form. Again, the pathways that regulate the detoxification mechanisms are upregulated. there is increased resistance to the various apoptotic, autophagic, and senescence pathway in the cells. And as I said, that there is an overexpression of the drug transporter protein, so the drug efflux mechanism is more strong in cancer stem cells. And specific stem cell transcription factors are activated. As a result, the cell can remain in a quiescent form, or it can divide to form one stem cell and one differentiated cell. So what are the key observations that say that there is an existence of cancer stem cell population? Because these cells are very few in number, we have seen that very small number of cells can lead to, on xenotransplantation, can lead to formation of the whole tumors. So even 10 cells will be sufficient to form the whole plethora of tumor cells. Whereas in case of the differentiated cells, that is not possible. So again, tumors grown from tumorigenic cells, they contain a mix of population which can be tumorigenic and non-tumorigenic. So the cancer stem cells are the population that are actually tumorigenic. These are some of the phenotypic characteristics that the cancer stem cells share with the normal stem cells, like cell renewal, cell cycle. The duration of the cell cycle is long in both stem cells and the cancer stem cells. Again, the genome repairability in stem cells as well as in the cancer stem cells is high compared to the normal cells. They are all located in the basal compartment, and the progenitor cells, they have short cycles to generate enough number of differentiated cells, and then they go back to a quiescent form or a dormant form. The same in case of the cancer stem cells. So again, they show plasticity, that is, they can differentiate and they can de-differentiate. Similarly, the cancer stem cells, they also can undergo change in the phenotype. Various environmental factors like radiation, oxidative stress, inflammation, changes in the environmental milieu can lead to transdifferentiation of a cell to cancer cell. So these in cancer cells can then undergo, these are, as I said, I'm talking about the epithelial cells or the solid tumors. So they can undergo a change from epithelial to a mesenchymal phenotype. So these EMT-induced cells are actually the cancer stem cells. So these cancer stem cells has an ability to move, whereas the cancer cells by itself, an epithelial cell, it remains stuck to its basal membrane. So by definition, epithelial cell has an apical membrane as a basal membrane. So cell is attached to the basal membrane. So when it undergoes an epithelial to mesenchymal transition, it detaches from the basal membrane and is able to transfer through the systemic circulation, leading to metastasis. In case of stem cells also, similar kind of environmental stresses can lead to mutation and formation of the cancer stem cell. So the cancer stem cells, they reside just like the stem cell within a small niche called the CSC niche. The CSC niche is isolated from the external milieu. And this is here where it remains dormant and proliferates. So once there are signals that mutation signals or stress signals, these CSCs, they exit from their niche and undergo differentiation to form the adult differentiated tumor bulb. So once the tumor bulbs are formed, they form the normal tumor. However, some of these cancer stem cells are retained as such within this tumor bulb, and then these cells can de-differentiate back. So this gives rise to the intratumor heterogeneity, and the cells, they can switch back from differentiated to a non-differentiated stage. So they can have a CSC to non-CSC phenotype. So that is what it makes it very difficult to isolate or identify the cancer stem cell, because it is not necessary that a cancer stem cell will divide to form a cancer stem cell itself. So the question comes is, are we actually targeting the right cells in therapy or not? Because as I said, that most of the drugs like cisplatin, paclitaxel, etc. are targeted towards the differentiated cells or the differentiating cells, which are actually the bulk of the tumor. But the few cells which are undergoing the epithelial to mesenchymal transition, or which we call as the cancer stem cells, may remain untouched. And once the bulk cell is removed, these cells persist. So over a period of time, these cells might come out of their niche and they can trigger the formation of tumor again. So that is the recurrence and metastasis occur. So therefore, we can say that specific therapies where we can target actually the cancer stem cells along with the traditional bulk population diminishing therapies can actually lead to improvement of survival and quality of life in cancer patients. And this is more relevant in case of a metastatic disease. So these are some of the various ways by which the cancer stem cells can be targeted. Like it can be done, as I said, that cancer stem cells are more chemo or radio resistant. So you can target the cancer stem cells so that the radio resistance or chemo resistant reversal can take place. The pathway interference can be done. They can be allowed to undergo differentiation. So they form the mature cells, which can be then removed by the traditional chemo or the radiotherapy. Then there can be disruption of the pro-tumorigenic microenvironmental interaction. As I said, that the cancer stem cells, they reside in a particular niche. So these also can be targeted. Then you can have anti-angiogenic or anti-vasculogenic therapies. Then you can have immune evasion. The cancer stem cells, they have that immune evasion pathways, so those can be targeted. And another thing is ablation through the prospective markers. Nowadays, for different types of tumors, the profile of the cancer stem cells are being identified. And once you know that whether this is the CD marker or extracellular marker that is present on the cancer stem cell, you can develop some antibodies against it and use the ablation methods. So coming back to some in vitro studies, so how do we isolate cancer stem cells? As I said that these are a dynamic population, so they cannot be targeted that this is a particular population. We grow them in the lab and we check out our cytotoxicity platform. That is not possible. So we look into the characteristics, like they have high efflux of chemotherapeutic drugs. That is what make them resistant to the drugs. So for separation of these cells, there are site population analysis assays and there are aldifluor assays. So this has been developed to isolate the particular set of cancer stem cells. Then as I said earlier also, the profiling of the cancer stem cells from different tumors have been done. And there are specific cell surface markers that have been identified for prostate cancer, for breast cancer, and so on and so forth. So we can use those expression markers to isolate those cells from the tumor bulk. Again, the cancer stem cells, they have a higher tendency to form tumors like spheroids, and they have the capacity to form the whole tumor by themselves. So again, you can have in vitro sphere formation assays and we can have xenotransplantation, where a very few number, even 5 to 10 cells can give rise to the whole tumor. So these are some of the techniques by which the cancer stem cells can be isolated. Though till date, the isolation of cancer stem cells from the samples remain elusive. So what we can do is, once we have a cancer stem cell population, we can enrich them. So this can be done, as I said, by specific stem cell marker, aldehyde dehydrogenase assays, and another important in vitro method of developing a model of cancer stem cell is through epithelial to mesenchymal transition. So as I said earlier, that through epithelial to mesenchymal transition, the cancer epithelial cell, it detaches from the basement membrane and it moves through the, it becomes more mobile and it moves through the systemic circulation to reach to the secondary site. When it reaches the secondary site, it undergoes again another transition that is called as mesenchymal to epithelial transition, and it establishes itself as a secondary tumor. So this is what happens during metastasis. So the cancer stem cells, they can undergo this kind of process. So one of the phenomena of EMT and identification of cancer stem cells are almost overlapping. So in our studies, we can generate a model of in vitro model of epithelial to mesenchymal transition and study the behavior of the cancer stem cells. So another method is by targeting the stemness associated signaling pathways. As I said earlier, that the the stem cell pathways like Wnt, Notch, Hippo, sonic hedgehog, these are all again these are again reactivated in a cancer stem cell. In our lab, we studied cervical cancer model. I'll be discussing some of these models, how we have generated the cancer stem cell population to study their behavior and target some drugs against them. Since loss of E-cadherin is a hallmark of EMT, and EMT is something that is closely associated with the generation of cancer stem cells, so we did an E-cadherin knockdown to generate cancer stem cells in vitro, just to check whether the cells which have undergone a loss of EMT are exhibiting the properties of cancer stem cells. We went in for some assays. So this is the knockdown model where the E-cadherin is low, and we found that concomitantly the expression of the mesenchymal cells, that is twist, vimentin, snail, were upregulated. So this shows that the cell is undergoing an epithelial, is losing its epithelial characteristic, is undergoing a mesenchymal-like phenotype. Now, comparing them with the cancer stem cell phenotype, we observed that there was an increase in the ABCG2 expression as well as the expression of the stemness marker OCT4. So these cells also showed high aldosterone expression. Another important thing is we saw that they have a better capacity to form in vitro tumorispheres. So that shows that by undergoing or inducing epithelial to mesenchymal transition, we have generated a population of cancer stem cell-like cells. Going for the functional activity, so we saw that these have showing a higher migration as well as invasive capacity, which is again a hallmark of a metastatic tumor. And as I said, that CSC-like phenotype, like OCT4, SOX, and NANOG expression was higher in these cells. Then next we went to patient-derived
Sorry, ma'am, I'm interrupting you.
Yeah.
Sorry, ma'am, I'm interrupting you. We have 5 to 8 minutes left. So I think you need to highlight the more important things. So, so at least
I'll take it. Yeah, I'll make it fast. So in the cervical cancer primary cell lines also, we saw a similar phenomena. So this is the tumor formation, and we did an immunohistochemistry for the developmental pathway in the patient samples, and we found an upregulation of both twist and GLI1, which are markers of mesenchymal as markers, as well as they are markers for the hedgehog. So we further looked into the therapeutic aspect. So in this case, we used hedgehog inhibitor, that is GAN58, and we observed that by adding GAN58, we were able to decrease the invasiveness of the cells as well as the tumor formation also was disrupted. The other markers were also down-regulated, markers of CSCs were also down-regulated where GNT was used. Cisplatin is the normal chemotherapy agent that is used in cervical cancer. So when we added the hedgehog pathway inhibitor, cyclopamine or GAN58, we observed that there was a marked decrease in the cytotoxicity of these cells. So this was also true in the primary culture, which we have derived from the patient cell line. Apoptosis also was enhanced when the combination of these drugs were used. So this is again, we did it also on the patient-derived cell line. So therefore, combining the conventional cytotoxic drugs with the cancer stem cell targeting agents can be more effective in case of cancer. So I won't go into the other models since there is a deficiency of time. Another thing we did was targeting autophagy. So this we did in urothelial cancer. So coming to the end, the targeting CSC alone is sufficient to in the early stages of cancer, but it is not sufficient for the tumor developmental period. So here we need to go in for the other traditional conventional methods. So combined use of cancer Stem cell targeting as well as the proliferating cell therapeutics may lead to a better effect during the tumor developmental period. And these are some of the therapeutic targets that are available that are already in clinical trial. We have LDH1, CD44, CD133, as I said, hedgehog pathway inhibitors. Then you have the CSC-directed immunotherapy also, the CAR-T therapy, which is an upcoming therapy for the cancers. They have been combined with the markers for the CSC. And this is a list of small molecular inhibitors that are targeting CSCs, which, as I said, autophagy, ferroptosis, hedgehog, notch, Wnt, hippo. These are specific targets that are available on the cancer stem cells. So by using this, we can target both the cancer cells as well as the cancer stem cells and eradicate the cancer population. So thank you. And this is my team. And there are not many people who were involved in these in vitro studies from different departments. So we have Professor Mandal, Professor esc-Singh, Professor Rashmi Bagga, and all these people from different departments and different institutes who have contributed to the data I have shown. Thank you. So the forum is now open for questions.
Yeah, thank you, ma'am, for your valuable talk. So we can take the questions now, please. You have any queries? We are here for more discussion now. We have 3 minutes left. So please, if you have questions, we can discuss on it. I have one question. So you said niche is very important for any cell. So how the normal stem cell niche is different from the cancer stem cell niche? So can you elaborate, ma'am, please, on this?
So as I said, there is an accumulation of many mutations that occur in the niche that lead to the formation of the cancer stem cells. So normally the stem cells are very quiescent. So they are, they do not they're resistant to all these kinds of stimulus. So whenever some kind of mutational changes accumulate, so basically the cancer stem cells are these are abnormal stem cells, you can say. So during the process of proliferation, some mutational change or some kind of stress has occurred, leading to disruption is the normal differentiation process. As a result, they stop at a particular state and they form the cancer. Whatever is cancerous is something which is indisciplined, that do not follow the regular biological principles. They do not follow the cell cycle. They do not adhere to the repair mechanism. So that is what is a cancerous cell. It's an indisciplined cell. So even in the stem cell, when the discipline is broken up, so it ends up being a cancer stem cell. So that is the concept. So it's a niche that protects it from becoming more indisciplined. So it's basically like you have a strict teacher who doesn't allow the indiscipline. So, but sometimes some people, some students, they just move away and they can cause chaos. So something like that.
Yeah, so one question is there, can stem cell repair cancer?
Well, stem cells have the most common and most effective stem cell repair mechanism in cancer is the leukemia. So you have bone marrow transplant. So it's not basically that stem cells are repairing cancer, it's basically replacing the cancer cells. So you remove all the cancer cells and you populate it with stem cells which can go and form the required type of cells. So another way where stem cells can be used to treat cancer is by using them as a bullet. So you can direct the stem cells to the specific sites. You can load them with chemotherapeutic drugs and they will reach the cancerous sites because they have an inherent, you can say, attraction to the wound. So that is why the cancer stem cells are present by nature in human. The residual stem cells in the living being is just to repair as a repair mechanism in case of emergency. So you have those cells within the body also as normal stem cells. So yes, stem cells can be used to treat cancer, as in leukemia. All blood cancers can be treated